Hey biologists, today we're going to be talking about cellular respiration. The equation for cellular respiration is the mirror image of the equation for photosynthesis. Our reactants are glucose and oxygen, while our products are carbon dioxide, water, and energy. An important part of cellular respiration is the organelle called the mitochondria. If you see here, the mitochondria has three parts. The first part is the outer membrane, this dark purple line. The second part is called the cristae. The cristae is seen here in red. Finally, anything inside of the outer membrane and cristae is referred to as the matrix. The first step of cellular respiration is glycolysis. Glycolysis occurs in the cytoplasm, which is outside of the mitochondria, but inside the cell. The process of glycolysis is the cutting of a glucose molecule into two parts. These two parts are called pyruvates. Seen here, we have one of the two pyruvates created from the first step of glycolysis. The second step of glycolysis involves a molecule called NAD+. NAD+ exists in the cytoplasm of the cell. When a pyruvate is created, NAD+ comes in and takes away electrons from the pyruvate. Now the pyruvate has no electrons and NAD+ has become NADH. It leaves to be used later in cellular respiration. Our pyruvate is now electron deprived and it is too large to pass into the mitochondria. To fix this problem, the pyruvate will attach what is called a phosphate group onto itself. This phosphate group will contain three carbon compounds. Well, three carbon which create a carbon compound. This carbon compound will break one carbon off, leaving two, so that the energy is created from this break-off and ATP is generated. This new molecule now has energy and two carbon. We call it pyruvic acid. This pyruvic acid is now small enough to diffuse into the mitochondria membrane. Once pyruvic acid has been formed, glycolysis is over. We are now entering the stage of cellular respiration called acetyl-CoA formation. Pyruvic acid diffuses into the mitochondria and then it diffuses through the cristae membrane. So it has now passed through the outer membrane, cristae membrane, leaving it in the matrix. Once pyruvic acid is inside the matrix, it will get rid of the energy electrons it was carrying in the form of ATP. It does this when NAD comes in, takes the electrons away so that it becomes NADH, and NADH leaves. The carbon compound has now lost its electrons. The last step of acetyl-CoA formation happens when a coenzyme attaches itself to these two carbon. This marks them ready for the Krebs cycle, which is the last phase of cellular respiration. The Krebs cycle occurs in the matrix. The first step of the Krebs cycle happens when a 4-carbon compound joins the 2-carbon compound to form a 6-carbon compound. Once this has happened, the coenzyme can leave because it has done its job. The 6-carbon compound has many new electrons. To help with this, NAD will come in from outside of the mitochondria and the cytoplasm, diffuse in, and the carbon compound will release one carbon. So now we have 5 carbons here and 1 carbon here. This carbon here has 2 electrons. The NAD will harvest it, becoming NADH, and now the carbon can be spat out as a waste product. We now have 1 of our 6 carbon as a waste product. The NADH will be moved into the electron transport chain. This process is repeated so that we now have another NADH inside the electron transport chain and another carbon outside as a waste product. When we have 4 carbon left in our 6-carbon chain, we use a molecule called FADH. FADH comes in from outside of this mitochondria and it harvests the remaining electrons. It becomes FADH2. This 4-carbon will now move outside of the mitochondria, and it too will join the 2-carbon and become, we will now have our 6-carbon waste products. FADH2 will move where NADH is. The electron transport chain occurs on the cristae wall. It's important to know that the inner membrane is in between the matrix and the cristae wall. When we have these electrons from NADH in the electron transport chain, proteins along the cristae wall will pump the electrons away from the NADH, creating NAD again, and it will pump them into the inner membrane. There are 3 proteins involved in this process. The first protein will pump them, the second protein will pump them, and the third one will finally push them into the inner membrane, but it will also use oxygen so that it can drop the low energy electrons and will finally form water. Once this water is formed in the inner membrane, it can leave the mitochondria through osmosis, and it will become another waste product, just like our carbon. Finally, the NAD can leave the electron transport chain because they can go back to collect more electrons to be used again in cellular respiration. Once cellular respiration has run its course, the last step of it will be ATP synthase, creating ATP, and it will push the ATP out, our last waste product, and we will now have completed cellular respiration.